Hi, everyone. Thank you for joining the second webinar in our series on the use of NAMs in risk assessment. This series is being co-organized by the EPA, the Physicians Committee for Responsible Medicine, and the PETA International Science Consortium. My name is Amy Klippinger, and I'm the director of the PETA International Science Consortium, and I'll be moderating today's webinar. We have two speakers today who will be presenting on dosimetry modeling of inhaled substances, Dr. Bahman Asgharian from Applied Research Associates and Dr. Rick Corley from Greek Creek Toxicokinetics Consulting. Before I introduce our first speaker, I wanted to point out that the slides from today's webinar will be available online at the link shown here on the screen. And a recording will also be posted there shortly after the webinar. If you're interested, slides and a recording of the first webinar in this series, which was on skin sensitization testing, are also available at this link. Following the presentations, there will be time for questions. Everyone is on mute now, but you can type any questions in the questions section within the GoToWebinar toolbar, which should be showing on the right-hand side of your screen. We will save all questions for after both speakers have presented. Speaking first today will be Dr. Bahman Asgharian. Bahman received his PhD in mechanical engineering from the State University of New York at Buffalo. His research includes biological modeling, cardiopulmonary fluid mechanics, aerosol physics, respiratory physiology, respiratory drug delivery, risk assessment from exposure to airborne material, and design and operation of inhalation exposure systems. He developed the Multiple Path Particle Dosimetry, or MPPD, software package, which has a graphical user interface to aid computational simulations. And today he will be presenting on the MPPD model and its applications. Bahman, whenever you're ready.
Okay. Hello, everyone. This is Bahman Asgharian, and I would like to talk about the multiple path particle dosimetry modeling that you have heard about. So I'll start my talk with an outline of what I will be talking about. Before talking about MPPD, which is a dosimetry model, I'd like to talk a little bit about what is dosimetry modeling, what are modeling approaches, and then I'll talk about the MPPD in specific, its features and applications. And by applications, I'm showing 3 examples to see how it works in practice. Then we'll have some concluding remarks. First, about dosimetry modeling. So as we know, LC50 or LD50 are not very informative. Instead, we would like to go to make a pathway-based risk assessment. That is to go from the source, which is exposure, to the biological outcome. And dosimetry models fill that gap here, as it's shown from source, which is exposure, to the target site exposure. So that's what the purpose of this is, to be able to complete this pathway. So what is the definition of dosimetry modeling in this particular application? It's mechanistic models based on physical and physiological parameters that govern the transport of materials in the lung. And using the model, we should be able to predict how much deposits, where it deposits, and what's the retained dose after initial deposition. So what are some of the benefits? It can complement or replace in vitro and inhalation studies. Modeling can be faster, cheaper, and also it can fill the gaps that we cannot do by inhalation studies, because can't do every scenario, but modeling gives us an advantage there. So it can also be used as a tool for interspecies dose extrapolation. And my third example actually will talk about that. So what are different dosimetry modeling approaches? In general, we have 3 approaches. First one is called site-specific. And by that, meaning we're looking at a small, isolated section of the respiratory tract, and particularly most useful in the upper airways, where we can reconstruct the geometry realistically based on the scan imagery. And the advantage, obviously, is that you can now, with this exact geometry, you can solve for the flow field and particle deposition. The second approach, which is called whole lung modeling, it considers the entire lung and entire respiratory tract, in fact. And then you are able to make regional and total deposition and those predictions. The third approach, which is the fairly new approach, combines the two, site-specific and whole lung modeling. Basically, what in this approach, for the large airways, we build the entire geometry from scans and solve the governing transport equation for the flow and particles precisely using three-dimensional computation. And for the missing airways, which is a deep lung, where you cannot really recreate the geometries only by measurements and the approximation, we do the whole lung modeling. And the two will communicate as one organic unit. So basically go back and forth. It's actually one unit. So how do you compare these three approaches? For site-specific, the strength is that we make no or very few assumptions, solve the flow three-dimensionally using Navier-Stokes equation, and you have either Lagrangian for particle tracking or convection diffusion equation for Eulerian approach. But the challenge, there are several challenges that limits the use of site-specific for the entire respiratory tract. First one is in calculating the flow field. The outlet boundary conditions are unknown. And what do you mean by that? The flow needs to know what goes in, what comes out. The outlet boundary condition actually is the pleural pressure that determines how the airflow is distributed. So if you don't have a good description of that, it doesn't do any good if you have really a strong tool such as CFD to make predictions. It's just not going to give you any very accurate prediction. It would be correct prediction, but it's not realistic. The other thing it needs is that you have to have realistic geometry. Use of a cylinder is not realistic. I mean, for an approximate model, such as whole lung model, is sufficient, but for the 3D CFD reconstruction, it is not, because if you solve a very precise flow in unrealistic geometry, how reliable is your results? And I have seen that some people, they break down the lung into sections and solve it sectionally. That is not actually an accurate approach, because in fluid mechanics, we know that all sections have to be solved together. One affects back and forth. So these are the limitations with site-specific modeling. Whole lung modeling, it makes sacrifices. It is able to solve and predict the position for the entire lung, and it's a useful tool for risk assessment, but requires assumptions. And obviously, the accuracy of the model depends on the assumptions we make. So assumptions have to be realistic and consistent with the level of accuracy we are seeking. And it also predicts average deposition per cross-sectional airway, and that's just inherent with the model assumption. Excuse me. So the third approach, which is hybrid model, is actually provides a framework for next generation of modeling, and it can include the emerging data information. It gives you the most accurate predictions to date. The only disadvantage, I say at this point, that we are building this. And next presentation, Dr. Rick Corley will get into details of how that can be done. So, oops, I'm backward. All right, so let's talk about MPPD. But before doing that, we have to realize MPPD is not the only software out there or the only model out there. There are some other well-known ones, such as NCRP and ICRP. So let's compare the three very quickly to see what are the advantages, disadvantages, and so forth. So in terms of the mathematical modeling, NCRP and RCRP are just semi-empirical. They are not science-based, basically. You fit data to a logistic equation, and that's your predictions. Multiple path, on the other hand, is deterministic, based on physics of the flow and particle transport. In terms of species, NCRP, ICRP, mainly humans, adults, and children. MPPD has that, plus it has other species, which I will talk about, and that's a good thing to have for interspecies extrapolation. For long geometry, all three models give you symmetric long geometry, but MPPD also gives you 5-lobe symmetric and asymmetric, completely asymmetric geometries. Particles, models can handle monodisperse and polydisperse. I haven't written here, but the MPPD can also do non-spherical particles, specifically fibers type or even nanoparticles, nanofibers. Breathing routes, nasal, oral for NCRP and ICRP, and MPPD can also do oronasal, normal augmenter, and mouth breather. It has also endotracheal breathing if needed. In terms of clearance, NCRP has 22 total compartments and 1 lymph node. ICRP has 3 fast, medium, and slow clearance pathways in the alveolar region. MPPD has a mechanistic model for the TB by mucus clearance and has basically similar to ICRP type model, but for each ventilatory units. Interface, NCRP is, you have to program it. ICRP does a window driven, and MPPD has graphical user interface, very easy to use, and has tutorial for a user to get up to speed quickly. So this is the structure of MPPD, basically. It models the entire breathing period from exposure to inhalation, transport in the lung airways, deposition, and clearance. So assume we have a size distribution. Inhaled it, a fraction gets into the upper airways. There you get deposition by impaction and diffusion. Some escape and get into the lower respiratory tract. In the TB region, you have impaction diffusion losses. And then when you go to pulmonary, you have sedimentation diffusion during inhalation. Then you have pause, where you have diffusion losses, Brownian diffusion for small size particles, and impaction for, sedimentation for larger size particles. And exhalation is a repeat of inhalation, except you don't have impaction anymore. So this is MPPD, what it looks like, the software. So you input your parameters, and then you make predictions. So let's see what are the inputs. First input, exposure characteristics. It's the mass number of particles. We have some uniform distribution, but it's not a restriction. It can be extended to any type of inhaled distribution. For particles, we need dimensions, size distribution, and correlation between length and diameter if you have non-spherical particles. Breathing parameters, which are basically breathing frequency and tidal volume. Breathing routes, I mentioned, is nasal, oral, oronasal, endotracheal. And we need lung parameters, such as FRC and URT volumes. And you also have the ability to make inhalability adjustments, depending on the scenario you're interested in. So the outputs, it gives you textual output, basically files that have all the input information plus predictions. You can also plot your results per generation, lobe, or region. It also creates data files if the user wishes to export to other graphical packages and spreadsheets such as Excel. So what does MPPD predict? It predicts the position for the entire lung, regional, per generation, lobar, and airway specific. And the position is made in human adults, children of different ages. Also in rhesus monkeys, guinea pigs, sheep, dogs, rabbits, and there are also mice and rats, which I have not included here. For retained dose, which is basically what remains after initial deposition as a function of time. So it can make airway generation for the TB region, and for the pulmonary region, it's just regional prediction. And we have retained dose calculated for human adults, for rats and mice. So with these predictions, what kind of matrix we can generate for interspecies extrapolation? It could be deposition fraction, deposited mass rate, deposited mass per surface area, deposited mass flux, and retained mass. Okay, now let's move on to the applications of MPPD. So there are three major applications. One is in risk assessment, which is the focus here, and there are two others for drug delivery and threat assessment. In risk assessment, it's used for characterization of exposure dose response relationship, for improving interspecies dosimetric adjustment and extrapolation. MPPD can interface with PBPK models to predict the fate of particles throughout the body. Also, it has the ability to actually serve as the pre step for inhalation exposure studies. So given that inhalation studies are expensive, you want to have an idea of what should be low, medium, and high exposure levels to produce certain outcome. So you can run the model, get an idea, and then use those to guide your inhalation studies. Drug delivery, it helps with drug formulation. And for threat assessment, it can predict the position of chemical, biological, radiological, nuclear agents in the lung, pretty much. So now let's look at three examples of how MPPD can be used. The first example, we'll look at the breathing route and activity level. So we look at the four breathing routes: oronasal, first nasal, oronasal, normal augmenter, mouth breather, and oral pathway. So we plot here the position fraction versus particle diameter from 0.01 micron to 100 micrometer. So we can see nasal deposition has the lowest and oral has the highest. And on this plot I'm showing you actually is in the TB region. And obviously the reason for that is because of the filtering in the oral or nasal cavity. And for submicron particles, since nasal airway doesn't, or oral airway doesn't play much of a role, so you get pretty much the same results. But for the coarse particles, you see the differences. And you can see that for fine particles, actually, if you look at now, go from light, moderate, and heavy, compare the three here. For the fine particles, not much difference. We can see a slight drop, actually. If you look at the peak here, you can see that drop with increasing activity level. And the reason for that is obvious, because as you increase your breathing, diffusion decreases. And for that matter, your curves drop. But for coarse particles, we're talking about here 1 to 10 or 100 microns, it's a different story. As you increase your activity level, you increase your flow rate, your impaction. So the position increases for large particles in the TB region, where impaction is significant. Now we look at the position in the pulmonary region. Curves are very similar. The pattern from nasal to oral are similar or the same, actually. But what we see here, that for fine particles now, it slightly increases as the activity level increases. So, and that is because of the filtering effect in the TB region. So you have less particles reaching the pulmonary region, so as a result, you have less deposition. For coarse particle, now actually it decreases. Before it was for TB, it was increasing. Here, the position for coarse particles are by sedimentation, not impaction. And impaction, when you increase your flow, your deposition increases. So that partly explains why you have decreasing deposition pattern with increasing activity level. But there is also another reason, and that is the filtering effect too. So the two combined causes a decrease in the pulmonary region when you increase your activity level. So now if you put them together for the LRT, so pattern's the same, but now the increase in the TB, decrease in the pulmonary, vice versa, the compensate, you pretty much get similar deposition slight differences, but they are not that different. So let's move on to the example 2, which is intersubject variability. So as we know, we all have different lung sizes with different breathing pattern. MPPD has about 30 stochastic lung geometries, which we can use to predict the position in each of them. And then when you plot them, you plot the position fraction as a function of particle size from 0.01 to 10 micron. You see a pattern that it resembles that to what you have seen before, except not for each subject is a different curve. The shape is the same. So you can see no variation, particularly when the particle deposition is at its peak, namely for diffusion and here for sedimentation and impaction. So, but in the middle, actually, there's less variation. We can also do that not just for the regional, for the lobar. You can see here for each lobe, you can do the same here. And right upper lobe has large variation, right middle has very little, right left has a fairly significant, and left lobe has less actually, given that they're all the same scale. So now example 3, which I think is of most interest for risk assessment. So we want to predict the human equivalent concentration using MPPD. We approach it 2 ways, for acute exposure or chronic exposure. Acute exposure, short-term exposure, the interest is in deposited mass. There's no time for clearance. So our dose metric should be based on deposited mass. And here we pick deposited mass per surface area. So how do you calculate deposited mass? Mass is basically deposition fraction, which is calculated from MPPD times the concentration of the material times minute ventilation times the exposure duration. So now, if you assume that the mass per surface area is the same between human and animal, now you can come up with an expression for the human equivalent concentration as a function of the animal exposure concentration. So this coefficient actually is the dose adjustment factor, which is used in RDDR, except obviously for the deposition fraction, because that is based on exposure, not deposition, for a particularly RFC methodology. So now we have a tool, and there's a simple equation to calculate the HEC. So here are some results in the TB and in pulmonary region. In the TB region, incidentally, this actually is from a work that we did back in 2005 and it's published in Inhalation Talks. So we are calculating the ratio of the human equivalent concentration to animal concentration. And we're going from 0.3 to 6 micron particle sizes. So our dose metric is the position per surface area, but this area is whether area of large airways, small airways, or all airways in the TB region. So the 3 curves actually are fairly similar in shape and slight differences. But what's interesting to note is that the ratio is larger than 1. So what does that mean? HEC over CA larger than 1 means that human equivalent exposure concentration is larger than the animal exposure concentration. So if you use the animal exposure concentration for risk assessment, in effect, you're underestimating the human equivalent concentration, and then you're overestimating the risk because you're using lower concentration for exposure. In the pulmonary region, you have a different matrix. Those metrics we can use could be mass per unit surface area, could be number of deposited particles per number of ventilatory units, could be per number of alveoli, or could be per number of alveolar macrophages. All depends on the dose metric of interest. And again, we plotted basically the same curve, the ratio versus particle size from 0.3 to 6 micron. So depending on what you use for your dose metric, you're going to get a different lines here. So that's key, obviously, what you pick for your dose metric to estimate your risk. So this is for HEC predictions based on the position or acute exposure. So what about when you talk about long-term exposure, which is a more realistic scenario? The position is not relevant anymore. In fact, you have to base your dosimetric on retained mass. And that makes things a little bit more complicated, not so much. The problem with retained mass is that the clearance is nonlinear. So you cannot derive a simple equation, which I showed a minute ago, between the human exposure concentration and animal concentration. So instead, you have to do a sort of a trial and error. Basically, you can do this in 4 steps. Suppose inhalation study has been conducted and we have determined what concentration animal reduces NOEL or LOEL. So we take that exposure concentration and run MPPD for the animal, the species of interest, and find the dose in animal. Now we make the assumption that dose metric, which let's say the position per or retained dose per surface area, is the same between animal and human. That's what we do in step 3. And now for this last step, we run MPPD again. Now we pick different human exposures until we meet that target dose metric. Once we reach that target, that concentration is human exposure concentration for retained dose. So let's look at some results. Okay, I've plotted same plots as for acute exposure, both in the TB region and in the pulmonary region. So I have the ratio of the human exposure concentration to animal concentration for particle sizes between 0.3 and 6 micron in the TB region and the pulmonary region. And we look at 2 scenarios. First, we assume that the exposure duration is the same between humans and animals. And also, we can assume that for humans, it could be a 70 years human exposure. So depending what you pick for your time, you get a different curve. The curves are similar in both cases, but you basically, when you use the same duration, you overestimate your human exposure concentration if you use that same duration. So what happens as a result, you underestimate your risk. Same true for TB and for pulmonary region. So that actually kind of concludes what I have to say, but now just close with a few remarks. First is that MPPD is a mechanistic model that is this great advantage. It's based on physics and physiology. And it has potential for extending to different particle types and species and so forth. It predicts the position fraction from which you can predict other dose metrics. So retained dose can be found in humans, rats, and mice. And for risk assessment, that's a valuable tool. So extrapolation, intra-species extrapolation can be made based on various dose metrics, which I mentioned earlier, the position fraction per surface area in TB, pulmonary region, or entire lung. And lastly, I want to mention that we realized that actually using MPPD to a specific scenario can be a little complicated at the beginning if the user is not unfamiliar with it, even though the tutorial is very easy to read and follow, and the learning is very, very fast. But we felt there is a need to include study cases in MPPD help menu for different scenarios we can think of. And with that, the user can just look at that, and based on that, just follow that and run it for the applications they have in mind. So this is an ongoing process, and hopefully we'll be completing that in a few months or so. So that concludes my presentation. I'll be glad to answer any question at the end of the presentations. Thank you.
Great, thank you, Bahman. And as you said, we'll have time for questions for both speakers at the end of the second talk. So please, if you haven't already, you can type any questions in the question section of your GoToWebinar panel. And also, as a quick reminder for anyone who joined late, the slides and a recording of today's webinar will be posted online soon. Okay, so speaking next will be Dr. I'm muting you for a second, Rick, you're giving me an echo. Speaking next will be Dr. Rick Corley. Rick received his PhD in environmental toxicology and veterinary biosciences from the University of Illinois. He is the sole proprietor of Greek Creek Toxicokinetics Consulting and a retired laboratory fellow from the Pacific Northwest National Laboratory at Oregon State University. His current research focuses on the development and application of multi-scale toxicokinetic models used to characterize target tissue dosimetry following exposure to chemical, biological, or therapeutic agents across species and life stage. Today, he will present on computational fluid dynamics-based dosimetry modeling of the respiratory system.
Unmuted.
Thanks, Amy, and also thanks, Bahman, for the setup. Hang on a second. There. So Bahman just covered a lot of details about MPPD and overall whole lung modeling in general. My talk is now going to focus in on the site-specific modeling that Bahman has discussed and some of the ways we've overcome many of the challenges for developing these types of site-specific imaging-based CFD modeling. And then I will end, as Bahman suggested, talking about the hybrid multi-scale modeling where we're combining the best advantages of either approach. So for those not familiar with computational fluid dynamics or CFD modeling, I just want to give a couple slides showing you basically what is it. And in essence, it's just a numerical method for describing fluid flows. And in this case, it's based on the Navier-Stokes equations that describe the flow of viscous fluids over a space and time. So you develop a flow velocity field based on your simulation condition using a computational mesh that has the right boundary conditions, such as what's the shape, what are the mechanical properties of the system, what are fluid characteristics, pressure, etc. And that's basically it in a nutshell. Then we overlay several other types of equations for the fluid flow to either interact with the shape or for carrying different materials over the geometry that you're describing. So for those unfamiliar with it, these methods have been widely used in a number of the physical sciences and engineering sciences, from aerospace, automotive, energy, building systems, and they've been used to help improve design, troubleshoot, and cut the costs in product development. So a lot of the tools and techniques in CFD models have had a lot of development over the past 30 years or so, and there's a wide user base of many of these projects that you can tap into. So unfortunately, the biological applications are more recent development. About 20 years ago, most of the CFD work dealt with heart function, blood flow, and fluid structure interactions. And the main reason for this is that these are the actual geometries that you need to create the computational meshes for these 3D flow fields are significantly more difficult to obtain in biological systems, as well as it's also difficult to obtain many of the boundary conditions that you use to drive the simulations. And lastly, and probably just as importantly, I think we still have a lot of math folks out here in biology, and for biologists to be working in this field, it usually requires some interdisciplinary teams and interactions. But having said that, both the respiratory CFD-based modeling and cardiovascular CFD models are a rapidly growing area with the advent of the new imaging technologies, image analysis, and computational capabilities that have grown over the past couple of decades. So let me go back to imaging-based anatomy. And what you'll see on your screen are CT scans from a rat, a rabbit, and a human. The scans are taking place from the tip of the nose, driving to the back of the nasal airways in the rat and the rabbit. And in the human, you're seeing the imaging stack coming from the top of the head, going through the head into the thorax, into the past the diaphragm. So these CT scans provide a lot of information. Airways show here in black, bone and pulmonary vasculature show in white, and then the rest of the tissues, the way we've got the CT set up, are showing in different various grayscales. And from this, we can extract airway geometries in the rat and the rabbit, and the human generally down to, in the human, 5 to 10 generations of pulmonary airways. And the rat and the rabbit, we can go a little bit further. If you'll note, though, on this human simulation, you'll see a lot of the white show up in the lung. Those actually are blood vessels that show up quite readily. And I'll come back to that point towards the end as we're talking about the hybrid models of why it's important to also see the vasculature. So over the past almost 20 years, the imaging protocols with our lab and other labs have been developed to where imaging in 3D as well as 4D by either magnetic resonance or CT imaging has significantly advanced the field. And over about a 10-year period, most of our laboratories, us as well as others, have worked on developing the image analysis techniques. We can go in and more rapidly pull out geometries of interest through a segmentation software that had to be developed, creating surfaces of airways or vasculature that you can then export into other software platforms for putting identifiers on the airway surfaces or vasculature for different regions of the airway system or cell types, and then creating these three-dimensional meshes that can then be used to run your CFD simulations. This used to take many months when we started this in the late 90s in our lab, and now many of these types of approaches can be done in really just a few days, and depending on image quality and what parts of the automated routines we can use, we can cut it down to hours depending on the quality of the data. So with this advent, I think personalized models are actually on the horizon. And most recently, 4D imaging has become another tool for us to use, where you can image over the breathing cycle or multiple time points in some of the clinical situation. And from that type of imaging, you can look at changes over time and extract airway and tissue mechanics through the entire breathing cycle that helps us improve the boundary conditions used in CFD model simulations. So at our lab, we have a suite of CFD-based models for a variety of species. If you need some geometry data to start working with, all you have to do is ask and we'll provide it. So I'm going to give a couple of examples of applications of CFD-based approaches. The first one being CFD with a PBPK boundary condition for reactive aldehydes. And the second will be a CFD aerosol dosimetry example for cross-species and in vitro comparisons. And then I'll end with the ongoing and future directions in multi-scale CFD whole-long aerosol modeling that Bahman has mentioned. So first example would be reactive aldehydes. In particular, it was these three aldehydes that we have worked with and developed models for. They're industrial chemical intermediates. Most notably, they're byproducts of combustion. They're also endogenously produced, or they have dietary sources. They are highly reactive, water-soluble vapors, and they're extremely difficult to measure in tissues. So when you do have inhalation exposures, what you generally see is contact site irritation, inflammation, exposure duration and concentrations are high enough, you can get cytotoxicity and degeneration. And then ultimately, over long exposures, compensatory tissue remodeling and even mutations. In terms of risk assessment for these chemicals, the cytotoxicity and tumors that are observed in nasal and upper respiratory tissues of rodents have been driving many of the human health risk assessments. Other effects, such as systemic effects, including leukemia, neurotoxicity, etc., following an inhalation exposure, however, remains controversial. So in purposes of our study, smoking is really one of the major sources for human exposure, and that's what drove some of our CFD modeling. And the other part of it is we can now, with the CFD, deal with the fact that we have an obligate nose breather in the rat, and the human, we have human nasal as well as the oral breathing. And in this case, for smoking, we can actually simulate the way a human actually takes a cigarette puff and following breaths. So development of the CFD models are all imaging based. What you see on the left is the nasal airways of a rat and the surface airway geometry where the individual cells for the computational mess are shown. Each airway cell has its own PBPK boundary condition, where the airway lumen that runs through the model is all driven by the CFD, but there is now carrying the aldehyde. You have diffusion of that aldehyde into the mucus layer and partitions in, continues to diffuse through the mucus layer into the epithelial cell layer where you can have metabolism or tissue reactivity, and in the submucosal layer. And one thing to point out with the aldehydes is the aldehyde dehydrogenase locations in the respiratory tissues is a key driver for tissue uptake and dose. It happens to be absent in the epithelial layer of the olfactory epithelium, but is present in the submucosal tissue in the olfactory region, whereas in all other tissues lining the airways, the aldehyde dehydrogenase is present in the epithelial layer. And that has a profound impact, actually, on tissue uptake and location. So we've taken these models, as I've shown earlier, and on the surface, we've identified where different cell types are located or different regions of interest that you want to have as outputs for model simulation. So it's helpful to preset your model with the sites that you specifically want to look for, or drive your boundary conditions. So these PBPK models have been set up for each tissue and cell type in both the rodent and in the human, and specifically with the cigarette exposures using the mouth exposures rather than the nasal inhalation exposures. So here's some results from the simulations. This happens to be for acetaldehyde at the rat no effect of 50 parts per million. So the question then becomes, in what type of dose metric should we use to compare across species and exposure? And to the left, you'll see different views of the rat nose and areas where you see surface fluxes of material that go across and into the tissues. And this was a technique that was used in the past, let's say, for formaldehyde exposures and risk assessments to look at surface fluxes. And you'll see the regions where the highest flux show in red and the lowest flux in blue. But by having a PBPK boundary condition, we can actually drive a little deeper and look at epithelial and subepithelial compartments and characterize tissue concentrations in more terms more familiar to toxicokinetics, such as area under the curve or peak concentrations. And here on the right side, I'm showing the location of peak area under the curve for tissue concentrations at the same exposure in different views of the rat nasal cavity, and then different slices through the nose corresponding to some of the main regions that are taken for histopathology analysis. And you'll see that the peak AUC tissue is actually the highest right in this area of the olfactory epithelium near the front part where the main airflow path comes through. And that is because of the location of the metabolic enzymes. In the olfactory epithelium versus the other tissues, it is in the submucosa. So it allows you to build up more of a tissue dose in the epithelial layer for this type of a compound. And as such, this area under the curve type of dose metric actually corresponds very nicely to the histology. And you'll see here's a cross section where you'll see a lot of tissue degeneration and remodeling from an aldehyde exposure in this dorsal part of the olfactory epithelium taken back here in about slice C. And that matches up very nicely if you do quantitative histology of various slices of the nose. The dose metric matches up quite nicely with where the histology is. So we use this AUC dose metric in a couple of comparisons published here below, and comparing what the dosimetry for each of these aldehydes might look like if a human was smoking a cigarette. And those human simulations were driven by a human smoking profile, looking at the puff, initial puff, followed by the several cleansing inhalation breaths after the puff, measured smoke constituents, the concentrations you expect to see in a given puff, and looking at, here I'm showing flux rates across the surface because it was a convenient way to output the model, but we are actually comparing AUCs of the actual aldehyde in the epithelial lining in its entire airway, and comparing those with the AUCs that we derived previously with the rat exposures at each of the no effect levels to make human comparisons with. And all those have been reported comparisons shown in this publication here at the bottom. So I'll move to the second example, which is with aerosol dosimetry, mostly for cross-species, but most importantly for the in vitro comparisons. This comes from a project with Syngenta that has developed an alternative method for re-registration of pesticides. In this case, normally the box checking approach for re-registration when you run short-term annihilation studies and you show lesions, and in fact with their particular pesticide re-registration, they were finding cytotoxicity and cell damage in the upper airways of the rat, mostly in the nose and into the larynx. And longer-term exposure studies, you'll get some lesions in the lung, but primarily you'll see the upper respiratory tract hit with cytotoxic aerosol responses. So rather than doing yet another inhalation study in rats for the re-registration for the risk characterization, instead of doing the 90-day study, they proposed an alternative method which involved in vitro studies in 3D cell culture systems, this case the mucilaire system from tracheobronchial epithelium in humans. They fully characterized exposure, both concentrations, particle size characteristics, etc., and various practices involved with worker exposures or for exposures for people that might get a spray downstream of where materials are used in mixers and motors and so forth. And then pulling it all together using CFD-based modeling approaches that can create a human equivalent concentration, exposure concentration relevant to the benchmark dose determined from the in vitro studies. I'll just point out the science advisory panel review of this proposed source to outcome approach was held December of 2018, and reports probably in the next weeks or a few months. But in essence, I'm going to give you sort of an overview of the CFD part of that project. And here I'm showing you a rat simulation at the concentration of aerosol, which is 4 milligrams per liter, which is a limit test in these animal studies, and the particle sizes that were used in the inhalation study at 2.7 microns mmAd. And on the left side, you'll see locations of where deposition does occur. And you'll notice that an awful lot of the material that's inhalable is deposited right in the anterior part of the nose. Roughly 58.2% what was inhaled is actually deposited in the nasal region itself. If you were to expose humans to this exact same aerosol size and concentration, you'll find that less than 1% is deposited in the nose for nasal breathing at a resting ventilation rate. Most of the rest of the material is actually taken down into the tracheobronchial and pulmonary region for this size of aerosol. So how do you go from that rat study and exposure study to realistic human and worker exposures and agriculture exposures? And through the characterization of the aerosols, most of the aerosols seen with use of the material range from 10 to about 35 microns mass medium aerosol diameter. And those exposures actually lead to very different sites of deposition versus the exposures determined from the rat study. So here you'll show 1 micron up to 30, just pictures of where aerosols are going. And below that is just a short table showing that the total deposited dose, percent of inhaled, in the nasal region through the trachea, which were the targets for the simulation. And it's not until you start getting to around 10 micron that you start getting appreciable deposition up in these upper airways. And then by the time you get to 30 microns, most of the material is up in the nasal passages and mostly in the vestibule itself. So this is another way of looking at that same data. This is a plot of total deposition, percent of inhaled, for each of these main regions. The nasal vestibule, obviously, as you get much past 20 micron, is taking up the vast bulk of material. And then these are the different tissue types lining those airways, where you'll see sort of a peak deposition in these upper airways. Once you exclude the vestibule, in that 10 to 15 micron diameter range. But interesting, with these site-specific models, the amount of surface area in these regions that actually receive exposure is quite low. And generally, well under 20% of the surface area actually gets a delivered dose. And even within that region where you have an area dose, there's quite a range in actually deposited dose concentration. So those become factors. Instead of looking at taking a total deposit dose over a whole region, you really need to be looking at deposited dose to the site-specific areas, because a lot of the surface area receives zero exposure. So if you go into the public docket on EPA's website, you'll actually see slides and all the presentations and backup materials for it, where they characterize the human equivalent concentration based on results from the in vitro studies, the particle size characterizations, and the CFD-based exposures. But I just wanted to focus right here today mostly on the CFD side of that equation. And once I think the report comes out, you'll see where there's any adjustments there in discussion on the human equivalent as it was presented by Syngenta. So where is this going now in the future? As Bahman mentioned, the current state of the art is to try to replace these overly simplistic airway boundary conditions, such as every airway outlet during an inhalation have the same pressure, with the idea that actually the airflow through those upper conducting airways are strongly influenced by what's happening in the lung that goes beyond the resolution of your imaging data. So I'm pointing out 3 laboratories that have been kind of wrestling with this problem for nearly 10 years, as well as our lab. The group at Iowa, where they've developed some multi-scale models based on 4D imaging. And the airways they construct generally about 5 to 9 generations on the CT scans, but they can get good ideas of what the lung shape and lung volumes are during the 4D imaging. And they appended their CFD models with volume filling, branching, an airway skeleton, so to speak, to help set the boundary conditions for their CFD simulations. Worthlaw and Jess group has also developed a multi-scale approach where he appends a stochastic individual path approximation, where one airway is essentially representative of all the other airways with their CFD models, and including moving wall models, especially for the ASNR region. And then Kleinstar's group has also been developing these whole lung models where the CFD is for the upper airways, and they're connected to essentially idealized trifurcation and further branching units in an ASNR region are appended to the CFD-based, imaging-based upper airway models. So all of these approaches are dealing with idealized versions of what's happening in the lung past what you can image. The approach that we've been working on, and Bahman is involved with this, is to really develop more of a personalized view of the airways past what we see with the imaging, and not rather than using an idealized approach. And we're taking advantage of a database we developed since the late 90s on 4D imaging, 3D imaging, and aerosol exposures in rats as well as human volunteers under both healthy and disease conditions, where we have imaging and aerosol studies done in the same individuals. And in this particular project, we are linking CFD and Lagrangian particle transport models with the multiple path particle dosimetry model, one-dimensional tissue mechanics models, and viscoelastic ASNR ODE models for each individual subject. We're incorporating imaging-based tissue mechanics for each individual, rat or human. And why I pointed out the vasculature in the lung, we're actually using the vasculature, which actually co-branches with your respiratory system during lung development, to develop personalized deep lung airway geometries for both the CFD as well as the MPPD configurations. In other words, every single airway outlet on the CFD model, the MPPD model is adjusted to that particular airway branching pattern based on the vasculature guide for how the branching goes for each individual airway. We originally proposed to also continue with the moving airways, but we've had some funding limits to get the whole project started. And so we may or may not be going down that path, but they are actively being pursued in other laboratories. Some of them are collaborators with us on our project. So this is sort of a graphical picture view of what this project entails. As you see on the left, we have the CFD model, or the airway geometry using the CFD model is from the CT scans of a human volunteer. And you'll see appended on every single airway outlet are these branch structures. That's the structure of the branching vasculature that matches with those particular airways. And that allows us to take these models deeper into the lung than the normal. So these branch structures are informing the MPPD model. And then the 4D imaging, and I'll show you that in a second, is used to extract airway and tissue mechanics. So we're replacing those standard assumptions, either uniform or zero pressure outlets, with these 1D and 0D models that incorporates airway resistance, compliance, pleural pressures, etc., based on the 3D and 4D imaging, as well as our measured physiology during these studies. So 4D imaging, this actually comes from one of the first imaging data sets we took with a rat that was on a ventilator, and 4T CT imaging over an entire breathing cycle. This rat had instilled elastase into one lobe of the lung to promote the tissue remodeling that's similar to what happens with COPD. And then the CT imaging are taking 11 times over a full breath breathing cycle. And you can see the movement here, but aerosol mechanics are implicit in this motion. And we can develop three-dimensional ventilation maps that are occurring right now as the animal is breathing, and carry that up to 3D. And from that, we can extract information on tissue compliance to incorporate into the model, as well as the measured physiology and respiration and the volume of the breathing cycle. We can start to determine some of the boundary conditions for pleural pressure at each region of the lung system in the rat. This same rat is also exposed to fluorescent microspheres, and what I'm showing on the left side here is the CT-based ventilation maps of the rat, and corresponding to the same cryomicrotone images of fluorescent microspheres deposited in the same animal. And when you plot the ventilation map versus the microsphere deposition, you'll find that it's highly correlated, which is what it should be, so that we can now look at where particles go from inhalation, as well as where the airflow goes in our development of 3D CFD aerosol dosimetry models, and compare those results to data in region-specific profile such as this. For the human studies, we had a number of volunteers, both healthy and with mild to moderate COPD, where we developed CT scans at 2 points in the respiratory cycle, FR functional residual capacity, and then add 1 liter, because that matched the exposures that we were going to also subsequently do with aerosol exposures with inert microspheres. And you'll see the picture on the left side showing the mask, where we can do either nasal breathing or oral breathing, and you'll see the imaging set on this particular volunteer, where you'll see the actual device. And each of these devices, at the very end of this, we have photometer probes that's measuring aerosols as they're moving through the exposure system into the mouth or into the nose, and then coming back out through exhalation exposure. And we're monitoring all the aerosol studies right now at the tips of these. So we include those devices in the CFD simulation. Studies were conducted by bolus as well as continuous exposure studies using this type of a system here, and with and without heliox. And exposures were all done at exactly the same supine position as the CT scans were taken from each of the volunteers. And here's one of the publications listed below for this study. So some of the early results for continuous exposures of 1 and 3 micron particles with nasal breathing versus oral breathing, the different subjects, there really wasn't a noticeable impact of the presence of COPD, mild to moderate, at this type of a breathing flow rate. And that wasn't entirely unexpected. But when we move to the aerosol bolus test, where you are introducing particles as a bolus at different points during the inhalation cycle, early, you're primarily ventilating the upper airways, and as you reach the peak inhalation, you're now starting to ventilate into the deep lung. The data strongly starts to tease apart those volunteers that have COPD versus a healthy lung in terms of what's exhaled. Because as you go deep in the lung, where you have tissue destruction, where diffusion becomes more dominant than the convective flows for deposition, as you exhale particles, you'll see a spread in the concentration profile over time as you exhale that type of under those disease conditions. So together, all these data sets are now being used to develop the models, as well as providing aerosol deposition information for model evaluation of its performance. So that's where the future is going, and this is just a summary slide for the CFD portion of today's webinar. I just want to point out that the time really has been cut down significantly how to develop these models with the new imaging techniques and all the software that's been developed now to deal with that type of data, as well as new techniques. And I didn't really have a chance to go into that. There's new programs out with high resolution omics that are helping pull apart function of different cell types along the airways. And I'll give a highlight out to the Lung Map Consortium that I used to be a part of before I retired from PNL, is developing those. And those provide important information on function of cells as they're along the airways of rodents and humans. But importantly, for this type of modeling, these are based on realistic anatomy, realistic physiology, and the physics of airflow and material transport. So that minimizes the type of assumptions and extrapolations from some of the more empirical or semi-empirical approaches. The resolution and exposure dose response can be significantly improved. That does present a challenge for validation, is do you have measurements of those a dose at that same level that your model can predict. Again, these models can be individualized to evaluate factors that control variability across subjects or on health status. In the end, we can significantly reduce the number of animals and improve experimental design to be more efficient and effective, much as what Bahman has expressed in the design of your future studies. As Bahman pointed out, and the projects I've summarized here, you can start to derive human equivalent concentrations for various points of departure from both either in vivo studies in other species or in vitro studies. And again, lastly, all the models we have developed, we do make available, so don't hesitate to ask if you have an interest. And then lastly, I just want to end with some acknowledgments of my colleagues back at PNL. I want to point out Chantal Darkane at UCSD, who's now taken over as the PI for this multi-scale modeling effort. And then of course, Bahman and his team at ARA, Syngenta for that source to outcome project, and colleagues back at University of Washington that we've been working with on the human imaging and the human studies. And with that, I'll conclude.
Okay, thank you, Rick. Oh, I gotta mute you. All right. Okay, so we now have time to ask questions to both of our speakers. Please do type your questions into the question section of your GoToWebinar panel if you haven't already, and we'll read them out loud. Before we do that, though, I wanted to turn it over for a moment to Gino Scarano. Most of you online probably know Gino. He's led the development of the strategic plan to reduce animal testing under the amended TSCA, and he's helping to organize this webinar series. Are you there, Gino? Well, let's give him a minute. He might be on mute. Let me ask one of the questions. So
Amy?
Oh, yep, there
Amy?
you are. Go ahead.
Oh, sorry. I went to hit the mute button and I shut the phone off. I apologize.
Okay, don't admit that on the live webinar, Gina.
That's fine. That's fine. You know me. Anyway, sorry for the initial mix-up in terms of signing in as one of the panelists. Thank you for introducing them. I want to thank both Dr. Asgharian and Corley for wonderful presentations. I'm sitting in here with 6 people, and I think we were all impressed and excited about what was said. So I'll let it go at that. And again, I want to thank PCRM and PETA for handling all the logistics in our joint webinar. So I appreciate it. And this is the second of what I know were going to be many webinars. So I'll stop there. I'll put it on mute with the correct button. And if anyone here wants to speak up, they can. Thank you.
All right, so I'll ask a couple of the questions that have been sent in, but do you know if anyone from your team has questions? Just feel free to jump in. So the first question I think that came in is for Bahman. What are the uncertainties associated with calculation of clearance with MPPD models for rodents and humans?
I'm sorry.
Oh, did you hear the question?
Can you hear me?
Yes, we can hear you now.
Okay. All right. As I said, the clearance models actually are purely semi-empirical model. Basically, you have a clearance rate constant that is a curve, basically, data fitted the logistic equation to clearance. And then we have a two or three compartmental model depending on the scenario. And so in terms of the uncertainty, that has to do with the data collected. The model itself is a simple empirical, semi-empirical model. I mean, in fact, if you go into MPPD, it has all the details. MPPD tutorial actually, and the content has all the details for the model, and there are publications on that as well.
Okay. So there's another question for you, Bahman. Interception is not included in the mechanism, ? Can you comment on that?
Sure. Interception actually is significant for elongated particles, not for spherical particles. For spherical particles, in a way, it is considered, I mean, even though it's small, in the numerical simulation, once the center of a particle reaches within a distance of a radius of the particle, it's intercepted, it's deposited. So in fact, interception and, the deposition mechanism are combined there. But for elongated particle, which was not the topic of this conversation, actually it is also included. It's there. But that's a different talk.
Okay. Let's see. We have, how is MPPD integrated into PBBK models?
Actually, for that, MPPD, I mean, we haven't done that. That has the capability of being done, but the way to do it is that you write your PBPK model and you call MPPD within that to predict those for the regions. That's, so if you're going to use a commercial software, I'm not sure how it can be, but if you write your own code, that should be pretty easy. Writing a code to do the PBPK modeling. So, I think writing codes for PBPK model are not that complicated because you're solving basically compartments and, basically ODEs, simple set of ODEs you're solving. So within that system, you just call MPPD, and that can, of course, the software it's not available, but that can be done theoretically. That's a more detailed discussion required, but I guess for now, I hope gives you some ideas how it can be done.
Okay. Okay, here's another one for you, Bahman. The publicly available version of the MPPD model doesn't have the capability to estimate particle deposition for highly volatile particles and particles in high number concentration. Do you have plans to add particle evaporation and particle cloud effects into the MPPD model and make the improved model publicly available?
Well, actually, that's a good question. I mean, you're correct. The publicly available model MPPD is just for inert particles, but we have other studies going on, particularly in the FDA. Actually, we're looking at cigarette smoke, which has all that effects that you mentioned, except the cloud effect, which we determined actually, while we thought it was significant in our earlier studies, turned out to be really more like a fudge factor. But in fact, if you account for the particle vapor interaction and absorption and all that detailed mechanism, temperature effect, that can explain the deposition of why, let's say, a cigarette puff, the deposition is much higher than what MPPD predicts. But all that, all that has been included in other. But if it's going to be publicly available, I don't know. Probably will, and it depends on FDA, So that's a different question, I guess.
Okay. All right, let's give Rick a question here. So Rick, what was the dose metric used for comparison between the estimated dose using CFD and the in vitro BMD?
Unmuted.
There we go. I was waiting to be unmuted. For that particular project, the dose metric, we ran a whole series of potential aerosol dose metrics. As I showed in the slides, the deposition, even within a small region such as the larynx or in some of the nasal tissues, is highly variable. You'll have some local hotspots of really high deposition versus areas of absolutely no deposition. So we ran a whole series of scales for dose metrics, such as total dose per surface area of the entire region that was of interest, dose scales within that region of deposition in areas where you actually had deposition, and what the dose per unit surface area was within a region, and we broke it into quartiles and minimum and maximums. So what Syngenta used for their comparison to the BMDL from the in vitro studies was the 75th percentile of deposited dose per deposited surface area of active ingredient, and that milligram per square centimeter 75th percentile was then used to compare against the in vitro BMDL levels, and simply running at this point, it's just duration of exposure, concentration of exposure that can lead you to that same BMDL at that 75th percentile dose metric for the regions of interest. In this case, it was within the larynx, corresponding to the primary site of lesions in the rat studies. So.
Okay. I think this question is also for you, Rick. Is particle deposition efficiency calculated from particle deposition count, or is it determined by each particle?
Unmuted.
It can go either direction. These are stochastic simulations, and we can looking at individual particle tracking. So we can tell you the numbers of particle per square centimeter. We can tell you the total mass. All those are part of the output from the CFD models. In the case of the prior question for the Syngenta project, they wanted to know the amount of material of active ingredient deposited per square centimeter, so they can correlate back to what they'd used in the in vitro studies. But that's part of the output. We can tell you a particle number, we can tell you a surface area, we can tell you mass.
Okay, and then here's a practical question, Rick. Can you comment on the practical aspects of the imaging techniques required for model building and development with regards to cost and required equipment, capabilities, and expertise?
Unmuted.
Well, all of the human imaging were done in the medical center at the University of Washington, and we've also received some other human imaging data sets from medical clinics from around the country. And so basically what it costs you to get a CT scan in the clinical practice is what it would cost us to get one as well to develop models from. It's nice though when you can actually work with the imaging specialists to tune the CT towards the soft tissue, such as the lung, as opposed to a bone. So there's several different ways you can kind of optimize the image, but the cost is similar to what it costs from a medical screening perspective. For the small animal imaging, we had our own CT scanner. So those laboratories that have those micro CT scanners, as long as you have the ability to work with animals in an environment with ventilators for live animal imaging, those facilities that have that capability can do those fairly routinely. If you are looking at high resolution or a high X-ray dose, then you're probably looking at imaging things like lung casts if you want to get that sort of imaging level, and we've done those types of studies also before. But right now we're using things that are in common with a veterinary school or a medical school or some of those labs that have micro CT imaging capabilities. And once you have that, you can usually contract with one of those to get the right types of imaging data sets that you need. Or you can ask us for what we've got.
Okay. So for the CFD Lagrangian model, can you describe the particle injection profile? Is it uniform or continuous?
Unmuted.
Okay, so for different types of projects, we'll do different types of injection type of profiles. For the Syngenta project, since we're dealing with really large particle size for the most part for the human simulations, we have a hybrid mesh where you have a prismatic boundary layer with a polygon core airway lumen. And we did random parcel injections within the core polygon lumen layer at the nostril entrance. So as the breathing profile evolved, you start the inhalation and the airway starts to come into the nasal passages. There are injectors spread uniformly within the airway lumen, and you'll have a random selection over the course of the profile that it's driven by the particle density, concentration, and the actual moment in time airflow characteristics that's being brought in with the CFD model. The reason we stayed within the lumen was some of these larger particles, if you put it in the prismatic boundary layer, they just weren't going to go anywhere. They'll just hit that wall right away. So we gave them at least a few millimeter head start before they ran into the wall at these larger concentrations. There's lots of other profiles you can use. We've set up simulations and run them where there's an atmosphere, a stable atmosphere of particles suspended in the gas, and we use the nose to capture and bring in particles that are minimal to the airflow as you inhale. Some of the pictures I showed had a sort of a cylinder in the front of the nose, and we've used that to sort of stage inhalation atmosphere. For the cigarette smoking, particle initiations were as a cloud in the mouth cavity, as a uniform distribution of the numbers of particles required to make the concentration and mass balance. And then as the breathing occurred with fresh air, that drew the mouth cloud into the conducting airways.
Right.
Now, I would point out that this isn't the sophisticated cloud that Bahman's been working on, where you have aerosol-volatile interactions going on and aerosol-aerosol interactions. So far, these simulations that I've shown you today, we're assuming there are no aerosol-aerosol interactions, aerosol-volatile interactions, or even aerosol airflow interactions. And at these larger aerosol sizes, when you start getting 10, 15, 20, 30 microns, and you get into very tight regions of the airways, these particle presence can actually influence airflow locally. But for the simulations we run here with these larger airways, we didn't include those assumptions.
All right, let's do a question or two for Bahman. Let's see, we have, is MPPD able to model hygroscopic growth of particles in the lung?
Well, the current version is for inert particles, but it can. And we do have versions for that. In fact, I published that back in 2004, but unfortunately, it's not in MPPD yet. So, but we can include it.
Okay. And can MPPD model be used for VOC?
For what, I'm sorry?
VOC.
Okay, actually, that's, we've done something basically for gases, vapors, and there is a version, but it's not publicly available. That, but I talked about MPPD, but we all know it's for inert particles. But I have articles, for example, for this, for the, volatile compounds. There's a 2001, 2012 papers I have, and has the details. But if somebody interested, we probably can work something out. But it's, if it's going to be, outlet, it probably won't be a part of MPPD. Would be a different, similar application, but with different package, because gases and particles are two different routes. It's two different, stories, pretty much.
Okay, and for MPPD, you have to enter a MMAD, but I have a VMD. So do you know the difference between the two? Can a VMD replace an MMAD?
VMD is that, I'm not sure what VMD, but MMAD is mass median aerodynamic diameter. If you use that in MPPD, you also have to calculate, input the actual density of the particle, because MPPD is based on the geometric diameter. So VMD the same way. It's if you have to perhaps convert it to CMD, MMAD, or MMD, which are inputs in MPPD. But VMD, I'm not familiar with. If you tell me what it is, I can perhaps tell you how you can convert it.
Okay. We can follow up on that after the call. Let's move quickly to Gino. I think you have a question from your end.
Thank you very much. I know we're nearly out of time. Go ahead, William.
I was wondering if any of the models can handle nanoparticle sizes that are insoluble materials but nano-sized.
Talking about MPPD here, nano-sized, soluble, or nano-sized particles?
Both.
Okay. For nano-sized, the current version of MPPD should be able to handle. And for nano-sized, particles, the earlier version did not have axial diffusion. So that's the major difference between, like, let's say, larger size particles. You don't have axial diffusion. Nano-sized particles behave in many ways like gases and vapors. So that's included. But soluble, we don't have it in there.
Okay. Rick, we have one for you. Can CFD predict retained dose?
Unmuted.
Okay. It depends. In terms of the gas and vapor exposures with the PBPK boundary condition, yes. In terms of aerosol at this stage, we do not have that included where we would add the clearance mechanisms. So if you could add either a PBPK boundary condition or say for particles, you can actually add a layer of mucus on the 3D simulation, and there's some publications where people have done that in the past, where you have a countercurrent motion of the mucus layer in the CFD side moving up the airways. However, those are computationally intensive calculations, and at this stage I haven't really seen anybody follow through with trying to invoke a 3D component to, say, mucociliary clearance. I think the way to do it going forward would be sort of doing the multi-scale approaches where you can sort of have an input-output going for different sites of the airways with a PBPK-like condition that includes the mechanisms, the various mechanisms of clearance, such as mucociliary transport, or in this case, some materials have solubilization as a clearance mechanism, or even macrophage or other immune cell, dendritic cell uptake as clearance. They can be added later, but I am not familiar with anyone who's actually done that with a CFD-based approach yet.
If I make a comment on that, actually, the thing is CFD and retained dose are two extreme ends, right? Because when you talk about CFD, it's very detailed, site-specific predictions, highly accurate prediction. Retained dose, you're talking about it's sort of a collection of particles, the net effect, what happens. But with CFD approaches, you don't have an alveolar region yet as of yet. That's something in the future, so you cannot do that. But for the trachobronchial region, mucociliary transport, there are ways to do it, but again, depends what level of accuracy do you want to do. And what Rick referred to was very detailed. But you can always put a simpler type, the type we have in MPPD. You can basically, in each airway, find out what is the mass and what is the average rate of the mass transport across different airways. And based on, you should be able to come up with a fairly high level meaning actually not very accurate, means high level of a high level model for the mucociliary clearance. But again, in the TB region, I don't know if there is a need to do that.
Unmuted.
Yeah, I would agree with that. I think you can do that multi-scale integration of the CFD in a model for mucociliary clearance in the TB region. I just haven't really seen anybody publish on evaluations of those types of studies yet. So Bahman and I are kind of working a review of these types of clearance models, and then hopefully if we turn up anything, we'll include those in the review. And it's also in some ways an out branch of the project we're working on, the multi-scale model development for aerosols.
Okay. Acetaldehyde is not a strong sensory irritant, so not much reduction in breathing rate from inhalation. By contrast, acrolein has an RD50 of 1 parts per million. How does this difference in effects on breathing rate get factored in?
Unmuted.
Yeah, that's a good point. The simulations that I showed you were all at no effect levels for the rat. I think the acrolein concentration was about 0.2 part per million for those simulation profiles. The key is you can use a measured ventilation profile. So if there are studies where you do have respiratory depression, we can use that as a front-end driver for the breathing, using that breathing measured profile in the CFD model. So you can handle it. We just used a resting ventilation profile in the simulations I showed because they were all done at no effect levels. But the RD50 studies done in mice, and all of my simulations were done in rats. So I'm hopeful that they were aware that they were below anything that's actually altering the inhalation profile. And these were also based on 90-day studies where those were the effect level came from, was the 90-day studies. We have published other studies where we've used different breathing profiles in rodents, such as sniffing behavior, resting ventilation, and then idealized types of simulations that compared the impacts of these. And you can get some modest changes in airway targeting in the nasal tissues of rats based on what type of breathing profile you use. And CFD models can use any type of measured or idealized profile you choose to use as your input.
Okay, I know we're about out of time here, but I'll try to run through these last couple questions really quickly. Bahman, going back to the earlier question, VMD is volume mean diameter. That was a question about, can a VMD replace an MMAD?
I don't think it can. Volume, if you know the density, you multiply by the density, it gives you MMAD. Okay, actually it gives you MMD, not MMAD. Because volume times density is the mass. So you have VMD, that becomes MMD, which is in MPPD.
All right, and then a question about whether the MPPD model can be used for e-cigarettes.
The current version publicly available, I mentioned that it highly underpredicts the dose, mainly because the vapor particle interaction is very strong. So you have three types, in an e-cigarette, you have three types of particles. You have medium, low, and high vapor pressure compounds. Medium is just tar in particle phase. High, such as formaldehyde, which is a byproduct, it's a pure vapor. And then you have like nicotine, and you have PG and glycerin, which are medium, they are in both phases. That interaction is key because the uptake by the wall tissues is both from the particle droplets and vapor. So that version actually we're working with the FDA will hopefully be out in maybe a year or so. But I would not advise using the current version of MPPD for e-cigarette.
Okay, and then last question. I think this one is for Rick. Is there modeling of absorption via the olfactory nerve or trigeminal nerve resulting in dosing to the brain?
Unmuted.
That was not included in the PBPK boundary condition. It's something that if you had the right mapping of those nerves onto the airway surface, like we've got essentially the whole olfactory region mapped to the airway surface, but if we could remap the neurons and the pathways to the brain, you could create a new PBPK boundary condition that could do such a thing. But at this point, no, that was not included with any of the PBPK boundary conditions we've used and reported so far.
All right, well, we're out of time. I wanted to thank everyone again for joining today's webinar, and to our speakers. We tentatively have the next webinar in this series planned for April, so please stay tuned for more details and a date for that, and enjoy the rest of your day. Thank you. Bye.
Thank you, Amy. Bye.
Bye.